These are the notes on biotechnology. Biotechnology, by definition, is manipulating organisms or parts of organisms to make useful products. And people have been doing this for thousands of years, actually. When we crossbreed plants or animals, when we make wine and cheese and beer and things like that, we're using biotechnology to do that. Nowadays, when we say the term biotechnology, we're actually talking about using modern techniques to look at DNA or other genetic material. And so nowadays, even though biotechnology includes those other things, it's really now today mostly about DNA technology. DNA profiling is something that is used quite a bit in biotechnology. It is using DNA fragments to figure out whether they came from the same individual or related individuals. Basically, what you're doing when you're DNA profiling is you're looking at non-coding parts of the DNA that have genetic markers that we can compare between individuals that show either the differences between individuals or different species or the similarities between them. And usually what we have to do in the process of doing this is amplifying or copying those markers for analysis so that we have plenty of them to look at. So here's basically how it works. Okay, you isolate the DNA, perhaps from a crime scene, as we see an example here, and also from the suspects. We're going to cut the DNA into pieces. We'll talk about that a little bit later. And we'll choose those markers. We're going to amplify the parts of the DNA that have the markers in it. And then we're going to put the specimens, all three of them, into wells in an electrophoresis gel and run that electrophoresis process, which involves running electricity through the gel, causing the DNA pieces to separate by size. And then we get a profile of bands we see on the pattern of bands on each part. And so when you compare the pattern of bands between these three different specimens, you can see that the crime scene DNA matches suspect 2 but not suspect 1. That's the basics of how it works. Now, to amplify the specimen, we do something called polymerase chain reaction, or PCR. This is a method by which we can copy a part of the DNA molecule. We use in the process of PCR, we have these primers that are short single-stranded DNA molecules that are chemically synthesized to match or to be complementary to parts of the DNA sequences at the end of each target sequence that we're looking at. And then we put it in this process. It's actually a 3-step cycle that each time you run the cycle, it doubles the amount of DNA. So here's basically how that works. Here's our target sequence over here on the left. You can see that this is double-stranded DNA. The first step in the process heats the DNA to separate the strands from each other. Heat separates the DNA strands because, remember, the two sides of the DNA molecule are held together by hydrogen bonds, and heat breaks hydrogen bonds. So that will separate the strands from each other. Then we have to add the primers, and they bond with the ends of the target sequences we're looking at there. We also add DNA polymerase and nucleotides to the mix, and the DNA polymerase adds the nucleotides in the correct order, and that duplicates the molecule. So now, after one cycle, we started with one molecule, now we have 2. The same thing happens with the second cycle. Now we're going to start with two strands, and then we'll end up with 4. And then at the third cycle, we'll have 8. Now realize you're not starting with one piece of DNA. You're starting with a bunch of DNA with those target sequences in it. And so it doesn't take very long to get a pretty big specimen of DNA from a relatively small one. Just in the course of a few hours, we can get millions of copies of those target sequences. And so it works pretty quick. It's kind of like a Xerox machine for genes. So this is a big advantage because it lets us take a very small sample and amplify it into a larger one. It doesn't take very long to do, and it copies only the part we're looking at there. Now we use gel electrophoresis to separate those DNA molecules based on size. So here's how it works. We have a porous gel that allows molecules to pass through. And of course, remember, anytime you have any kind of a porous substance, smaller molecules, smaller parts are going to go through faster and more easily than larger ones, okay? So basically, you put your DNA specimens, the DNA pieces have been chopped into fragments of different sizes. And so when you apply the current, the electric current, remember that Electrons, the electricity is the flow of electrons, negative electrons, from a negative pole to a positive one because they're repelled by the like charges and attracted to opposite ones. DNA fragments are also negatively charged, and so they're going to be pushed along by the flow of electrons toward the positive electrode. The shorter pieces are going to move through the gel faster and farther than the larger pieces are, and so they're basically going to separate out into bands that are more or less the same size. And so what you'll end up with after you stain it or look at it with fluorescence or radioactivity, depending on the method you're using, you'll end up with a pattern of bands. Each band would be a collection of DNA molecules of the same length, and the pattern that is discerned there is different for every person, unless they're identical twins or clones. So here we have, again, we have a mixture of the fragments that are added in there. As we run the electricity, it moves those fragments. The shorter ones are farther away from the origin, and the longer ones are closer to the origin there. So the shorter ones are faster, the larger ones are slower, and you'll end up with a pattern. Then you can match the pattern to see the similarities and differences between them. We use DNA profiling for lots of things these days. Of course, it's used a lot in court cases to determine guilt or innocence, or perhaps determine paternity of a child. You can also use it to identify victims of accidents. You can also look at other materials, non-human materials, and look at origins of those kind of things. We can look at various different species and see how similar or different they are. We can look at wood or any kind of plant material, or anything from living sources that has DNA. We can use DNA profiling to see similarities and differences. Now, another process, or when we compare those, what we're doing is something called restriction fragment length polymorphism, or RFLP. The single nucleotide polymorphisms are differences at a single base pair within a genome. Sometimes you have a substitution that's wrong. Sometimes you have a base pair that gets flipped over. And so what that can do is it changes the length of the restriction fragments, or the pieces of DNA, because it's going to sometimes changes the restriction site or the recognition site for the restriction enzyme. So we're going to produce the DNA fragments using restriction enzymes, and then we're going to sort those out by gel electrophoresis. So here we have an example of a SNP, or a single nucleotide polymorphism. Here's one base pair that got changed here. The C and G got substituted with an A and a T. The particular restriction enzyme that we used in this case cuts with a zigzag cut between the C's here. And so you'll see in the original sample, we've got in the wild type, I guess we'd call it, we have two cuts, which makes a total of 3 pieces. But in the second sample, the mutant one, or the one that's different, notice that since that's changed, we can't cut between the C's there. It doesn't work that way, and so we end up with only 2 pieces, and so that will give us a different profile from that particular one. And that's one thing that makes our DNA profiles different from one person to the next. Okay, here we have a couple of DNA profiles, and you'll see several of these on your test, of course. This is a paternity case over here. The standard that's listed here is just basically a ruler that tells us how many base pairs long the different bands are. So here's our child's DNA. Remember, every band in the child is going to match up to one in either the mother or the father. We know this is the mother, so you can see the bands that match here with the mother. And they're trying to determine whether this man could be the father of the child. And you'll notice that the bands that the child has that did not match to the mother match one from the father, so he very likely is. In this case, we have a crime scene with a couple of different suspects here, and we can tell easily that the crime scene DNA profile matches the DNA profile from suspect number 2, which means that that person, suspect number 2, is the person that left the DNA at the crime scene. It does not prove that suspect 2 is guilty. It proves that the DNA that was recovered there is his. The rest of the proof of someone's guilt has to do with other things besides just that. That's just one more line of evidence in the proof of whether someone is guilty or innocent. Genomics is what we study when we look at the complete set of genes and their interactions in organisms. The first ones that were studied were actually prokaryotic genomes. They're relatively small compared to eukaryotic ones. Since that was first done back in probably the 1970s or 80s, a lot of eukaryotic genomes have been investigated since then. This lets us look at evolutionary relationships. This is how we know that we have a 96% similarity in DNA sequences between chimpanzees and humans. We can also figure out functions of disease-causing genes because we can compare human genes to similar genes in yeast and other kinds of organisms. And so this lets us see similarities and differences between disease-causing genes, between individuals, between species. There's a lot of useful things we can get from the science of genomics.